Dr. Anjali Venkateshwaran, and we'll be introducing today's speaker on behalf of Biomir, a preclinical CRO with locations in Boston, San Francisco, and China. Welcome to the webinar series on novel approaches to cell and gene therapies. Our speaker today is Dr. Casey Maguire, an associate professor of neurology at Harvard Medical School and an investigator at Massachusetts General Hospital. Casey received his PhD from the University of Rochester Medical School. He has been at Mass General Hospital since 2006, starting with a postdoctoral fellowship, followed by instructor and assistant professor positions. His research focuses on improving virus vectors to allow effective human gene therapy in the face of delivery and immune-related barriers. Casey's talk is titled Strategies for In Vivo Barriers to Gene Therapy Vectors. So without further delay, it is my pleasure to introduce Dr. Casey Maguire.
Thanks a lot, Anjali, and I want to thank Biomir for the chance to give this talk today. Again, as Anjali mentioned, I'm going to be talking with you about strategies for in vivo barriers to gene therapy vectors. I'm going to be covering a lot of data from our group, but also a lot of pioneering work from many of the other scientists in the field. Unfortunately, due to time constraints, I can't give everybody proper credit, but I'll do my best. So this is my disclosure slide. Again, due to time constraints, I'm not going to go through this, but it'll be available for you to look at if you need to. To begin, I'm going to give you a little background on gene therapy. So the basic goal of gene therapy is to correct a disease by addition of a normal copy of the gene to compensate for non-functional or the absence of a gene product. This is typically done with recessive mutations, and this is often called gene addition therapy or gene replacement therapy. The second thing that you can use, another strategy of gene therapy, is editing or deleting a mutated gene. This is often in the case of a dominant mutation when there's a pathological effect of the gene product. This can occur, this can be done with gene editing with CRISPR, TALEN, zinc finger nucleases. It can also be done for recessive mutations if you have a donor DNA and you can actually repair the mutated gene. The third strategy is altering expression levels of gene products. This can be done with siRNA or artificial microRNA when you actually reduce mRNA levels to alleviate some of the pathology of the disease. Finally, you can add in exogenous genes, sometimes genes from bacteria, bacterial toxins. This is used in cancer gene therapy, or overexpressing cytokines to immunomodulate. I also wanted to give a little historical timeline of gene therapy, just to give some context of how much has been done and how long the field has actually been around. A lot of folks think of gene therapy being around since the 90s, but really, when you look back at the literature, it really got its roots all the way back to the 1960s, when scientists began to understand how viruses such as SV40 could genetically modify cells and transfer their genome into mammalian cells. Again, this isn't meant to be comprehensive. I'm going to try to cover most things related to adeno-associated virus, or AAV, as that's the focus of my talk today. So AAV was actually discovered way back in the 1960s as a contaminant in adenovirus preps. And in the 1970s, Friedman and Roblin actually conceptualized gene therapy as a way to treat human disease. The first retroviral vectors were made in 1981, and in 1982, Jude Samolsky in the lab of Nick Maziska actually cloned AAV into a plasmid, kicking off that field. In the 1990s, there was the first ex vivo gene therapy trial in humans. That was with a retrovirus vector. In 95, it was actually the first AAV gene therapy trial for cystic fibrosis. Now, the field hasn't been without its setbacks. In the late 1990s, early 2000s, there was an actual serious adverse event with an adenovirus vector in a clinical trial, followed by another serious adverse events with a retrovirus vector for X-linked SCID. So that caused a kind of a pause with the researchers in the field to really think about what vectors were best for what indications, and how to build safer and more efficacious vectors. And that's just what these pioneers and scientists did. So coming up through the 2000s, we really started to see positive efficacy in clinical trials. One example is AAV2-RP65 for Leber's congenital amaurosis, which is a rare blindness. Also, there was good clinical findings for factor 9 to treat hemophilia. And then starting in 2012, both in the EU and the FDA approved 3 different AAV-based gene therapy products. So now AAV gene therapy, or gene therapy in general, have moved from this sci-fi, great theory into a clinical reality with medicines being used in people today. So first, I'd like to give you some background on adeno-associated virus vector, the virus vector that we work with in our lab. It's a small non-envelope virus, about 20 to 25 nanometers capsid, has a single-stranded DNA genome. The wild-type virus has rep and cap genes. Rep stands for replication. There's 4 of those proteins, and cap, that stands for capsid. That's the structural proteins, VP1, VP2, and VP3. There's also some newly discovered genes within AAV, but I'm not going to go into that today. The insert capacity is about 4.7 kb. And with a conventional AAV vector, you can basically take out those rep and cap genes. It's completely gutted, meaning you take out all those genes, you provide them in trans in the producer cells. Therefore, you make room for your gene of interest, where you have your promoter and your transgene. That's shown up here below the capsid. The only cis sequences that are needed to be packaged into the capsid are these ITRs. They're inverted terminal repeats. They're needed for packaging and replication in the producer cells. But the vector itself is completely replication incompetent. The vectors have been shown to give long-term extrachromosomal transgene expression in differentiated cells, such as muscle, neurons, and liver. The vector can be diluted, meaning you can lose transgene expression if the cells divide a lot, such as in tumor cells. Although using different strategies, such as genome editing, when you only need a short-term expression to make permanent modifications, AAV may still be kind of the right choice for that indication. As I mentioned, there's been efficacy in many different clinical trials, and there's 3 FDA-approved AAV-based medicines. However, there are current limits of AAV efficacy in these vectors. Really, that relates to treating a broader base patient population. We want to treat more people, make more patients that need this therapy available for treatment. So there has been reports of dose-limiting toxicity. I mean, this is likely related to the transgene overexpression. It's not always observed, but it's been seen in some non-human primate studies. The relatively low efficiency of CNS transgene delivery after systemic injection, so that's a barrier to get really more efficient expression in the brain when you want to use the IV route. There's immune obstacles. That's probably the biggest obstacle right now that we face. There's CD8 T cell responses against the capsid, which can eliminate transduced cells. There's also pre-existing antibodies when we get exposed to the virus. There's also transgene capacity. Again, it's about 4.7 kb, which fits many genes, but not all of them, although I'm not going to have time to cover this limitation today. So again, I'm going to be talking about a lot of strategies around evading the immune response. So I'm going to cover that here in a little more detail. So a lot of us are exposed to wild-type AAV, just the virus, early in life. And then we mount an immune response to that. That leads to anti-AAV antibodies and capsid-specific T cells. Now, when we give the AAV vector for gene therapy, our body recognizes that as the wild-type virus. This leads to the humoral response to the capsid. The antibodies are already circulating. So in some patients, they're actually excluded from clinical trials. There's also a cellular response to the virus capsid, as I mentioned, and that can result in elimination of the transduced cells. This was seen in hemophilia trials. Now, this doesn't happen in all patients, and there's strategies such as high-dose steroids, which seem to mitigate that to some extent. You can also have immune response to the transgene product, which can also limit the therapeutic efficacy. If you can, if the patient doesn't have these issues, or if you do have to go around these, the end result is long-term gene expression. Also related to CNS delivery, there's of course the blood-brain barrier. So typically what's been done to get around that barrier is just to directly inject AAV into the brain. This does have a lot of attributes, such as you get low expression in other organs, so you reduce chances of systemic toxicity or immune system activation. However, you do have less greater spread of the virus. So for diseases where you really need to hit a large number of cells, this can be a limitation. The other option is to use a systemic dosing of AAV that viruses that can cross the blood-brain barrier. And amazingly, in 2009, it was discovered that AAV9 could cross the blood-brain barrier after IV delivery. This has been shown to work in mice, pigs, monkeys, and even humans now. You get good spread of virus in the brain, although the efficiency can be improved a lot. But you also get high expression in heart and liver if you don't use, restrictive promoters. So this can lead to immune activation and toxicity. So today I'm going to talk really about two different approaches that we're using to overcome delivery and immunological barriers. The first is an AAV capsid library to select for vectors with improved CNS delivery. And the second is using things called extracellular vesicles to enhance AAV-mediated gene delivery. So what are capsid libraries? They're basically, you can clone randomized ligands of a capsid, or you can make these capsid chimeras. I'm going to go into a little more detail in the next slides. You can use high efficiency cloning techniques, and you can make diverse libraries with 1 million to 100 million or so independent clones of viruses with these different ligands. And then you perform in vitro and more commonly in vivo selections, which basically result in enrichment of clones, which can complete the entire transduction process or other phenotypic traits that you want. So as you can see in round one here, you have a mixture of different capsids represented by the different colors. As you go through the subsequent rounds, maybe you're selecting for a vector that can go to the brain. You can see the screen virus has basically outcompeted the others. It's survival of the fittest, and you can select for efficient vectors that way. So peptide libraries have been used for probably 20 years now by many different groups. This is just an example shown here just to illustrate how it works. You're basically cloning random peptide inserts into this exposed region of the capsid at the threefold axis of symmetry. You can see these 3 blue dots all over the capsid, and that's a trimer of the peptide in the VP3 region on this exposed loop region of the capsid. There's also these so-called DNA shuffled libraries, which again, this technique has been around for 30 years or so, but folks started using it for AAV some 15 years ago or so. In this approach, you basically are going to take several different full capsid genes of AAV. This is represented by the different colors here. You can use DNAs and chop up the capsids into little bits, and basically through PCR reassembly, you're going to make these really chimeric capsids that basically are comprised of multiple capsids, shown here with the multicolors here, and that will give you capsids with different biological properties. And then you do the same type of selection approach that I showed you earlier. So some key factors to consider when you're performing AAV library selection. First is considering the actual library complexity or bias or parental virus contamination or defective capsids. These are all things you need to be aware of when you're first starting out working with these libraries. The selection approach, this is really critical. And finally, choosing your candidates, that can be a really important step when you're doing these selections. So what am I talking about when we're talking about actual library complexity or bias? So the theoretical complexity, I'm going to give you an example here of a 7-mer amino acid library. There's 61 codons, and you're going to have 7 positions for those amino acids to be placed in the 7-mer library. So that will give you about 3 trillion combinations. That's a lot. However, we have a degenerate amino acid code, so many of these codons encode for the same amino acid. So there's only 20 amino acids that you can code for. That's still, in 7 positions, give you a billion theoretical combinations. That's still a lot of diversity. However, when you're talking about actual complexity, this depends on several factors. Again, the degenerate code, so you could have unequal representation of some amino acid because there's 1 to 4 codons per amino acid. The technology used to make the library, a lot of people use these NNK primers that allow you to remove some of the stop codons and some of the degeneracy of the codon to give you more equal representation. The process of plasmid generation prior to the AAV production, we're going to talk about that more. And really what peptides work in the context of the capsid. You may have good diversity, but when you go to make the virus, these viruses can be have to form properly to be functional, and depending on the function of that peptide, that can disrupt the capsid. So here's some of my tips based on some of our work and really what's been found in the field for maximizing library diversity. First is wild type AAV contamination. The possible solution for that, that happens when the plasmid used to make the library has some wild type AAV9 or whatever serotype you're using. Or it can be, if you're working in an AAV lab, there's a lot of in the hood, or there's a lot of plasmid from other preps around. So you can use restriction sites that kill wild type AAV capsid, but not the library. This was done by the Gradinaro group and has been a really good trick. You can use dedicated reagents, dedicated incubators, and hoods for library production. Really try to isolate your library production from your standard AAV productions. You want to maximize complexity. To do this, don't transform into bacteria. You can directly transfect the ligation. This has been done again by the Gradinaro group, Deverman et al. Because when you grow up in bacteria, you can actually, reduce your complexity for certain sequences. You can optimize your PCR with randomized NNK primers. You can reduce the codon-rich amino acids. Again, you can also use available semiconductor-based technology from CROs to provide precise control of amino acids and coverage. So making sure that there's all the amino acids across each position. So to minimize defective capsids, you really want to minimize the cross-packaging by using low numbers of plasmids per cell. I'm going to talk about that in a minute. You can minimize these stop codons again by using the NNK primers. There's 32 codons, and you can eliminate 2 out of 3 of the stop codons using that. Or again, you can use the newer library technologies that I mentioned above from CROs that can give you better diversity. Finally, the selection approach here is really important. It's a critical step in optimal use of directed molecular evolution. The key thing to remember when you're doing these studies is this is a black box approach. We really aren't taking it from, we're not targeting a specific receptor. We're asking the viruses to find the best route to give you your phenotype. So you get what you select for, even if it wasn't what you wanted. You have to be really careful about the different parameters you put into your selection schemes. So just to remember that the selective pressure really drives the condensation of the library to select for a smaller number of capsids, because you really can't individually test 1 billion capsids. That's why there's a selection approach here. So you really want to think carefully about the selective pressure of your model. You want to do you want to select in one strain of mouse, or do you want to select across species, or do you want to really go to a more clinically relevant species, such as a non-human primate? This has been really shown to be an issue over the last several years, where some of these vectors you select may work great in mice, but they don't necessarily work in another more relevant model, such as non-human primate, possibly because some of the receptors are conserved or not expressed in the larger animals. So does your model's physiology in the target tissue represent your desired phenotype? Should you select a naive brain if your target is in an inflammatory environment, such as Alzheimer's disease, and vice versa? Are you inadvertently putting inflammation into your model where you don't want it? So choosing candidates, this is important things to consider. First, both the complexity of the initial library, The capsids that can produce well can give you overrepresentation. They may actually not be giving you really great function on what you want, but they just replicate and package really well. And you get selective pressure. That factors into how many candidates you have at the end. So usually candidates are chosen from NGS sequencing data. Historically, this was Sanger sequencing, and you really had you didn't really have a good idea of the true depth of the libraries. Now NGS is a double-edged sword. You now can see really a lot of what's in the library. So sometimes it can make choosing the capsids a little more tricky because you have a lot more to choose from. So some tips are, you got to perform multiple rounds to weed out the less efficient capsids and enrich the most efficient. Use a strong selective pressure, such as transgene expression. I'm going to talk about that in our example from our lab to condense, and that's used to condense the library in fewer rounds. You want to follow the NGS data for the most enriched capsids over rounds 1 to 3, sometimes versus just the most prevalent clone, because you may be able to find, this may show you that you're not, that the most prevalent clone can just replicate well. You really want to look for a big jump in from round 1 to your final round. So what is cross packaging, and how does that lead to problems when you're doing these selections? I'm just going to give you a little diagram here, a simplified diagram. So there's loss of genotype-phenotype linkage, and this can confound selections. So you have your capsid genes for a blue capsid and a green capsid here, and they encode for these blue and green capsids. When you produce these and package these in your producer cells, the blue DNA will make the blue capsid, the green DNA will make the green capsid, but also what can happen is the green capsid can package the blue DNA, and that leads to that genotype-phenotype disconnect in the linkage. So when you do the selection, and let's say the green capsid is really good at going to the brain, you do the NGS, which relies on sequencing of nucleic acid or the DNA, you'll think the blue capsid was the best, which will be wrong. So really, it was the green capsid, and to get around that, as I mentioned earlier, people have just found there's a sweet spot where you can use lower amounts of DNA so that this cross packaging doesn't occur in the cells. You just mitigate that there won't be too many AAV genomes per cell. So I'm going to talk to you about now an example selection from our lab that we recently published. So an AAV vector has to go through many steps for efficient transgene expression. The first is distribution to the target organ. Then you have cell binding, internalization and intercellular trafficking, nuclear import, and there's several other steps there too on coding, second strand synthesis, and then finally transgene expression. Most selection schemes don't differentiate capsids outside the cells, inside the cells, or within nuclei. The Gradnaro lab is one exception to that, where they look at capsids that go to the nucleus. So we thought that selecting at this step, transgene expression would be optimal. So we call this the iTransduce platform, and what it is you have a conventional AAV9 capsid library with 7 mer inserts, and then you also have this sensitive Cre recombinase cassette. You can drive that with any reporter. And you do a library injection, similar to any strategy. You can do any route of administration. We were interested in IV delivery, getting capsids that go to the brain, and use this flock stop CD tomato mouse. And if the viruses can't mediate transduction, you get no Cre, and you get the stop transcription signal isn't removed, and you don't get fluorescence. However, if you have Cre expression with a functional vector, you get the stop signal is removed, and you get TD tomatoes expression. And you take out your organ of interest, in this case it's the brain, and you can dissociate that, and then you can look for these fluorescent cells and sort those instead of sorting a whole population of cells with a lot of probably defective capsid, and then you isolate the DNA from those transduction competent or fluorescent cells, and select your candidates from that. So we did a selection. We did 2 rounds of selection. The first round, we did a conventional round of selection to condense the library. And then in round 2, we basically did either the iTransduce selection or a conventional selection, really to compare the peptides that resulted from that. So we did NGS on the round 2 samples from either the whole brain. And what you can see here, this is just a donut chart showing all the peptides that we obtained. All the little blue lines are individual peptides, and this is just a ton of other peptides here. But you did see some significant enrichment of these 2 capsids. With the iTransduce selection, we really only had 2 viable 2 capsids identified, and this one had the stop codon, so we didn't pursue. It's probably due to cross packaging. We did see this one capsid, this FEV, show up in both the round 2 conventional selection and the TD tomato selection, which also gave us some confidence that this was a viable peptide to go after. So we basically just made test capsids with a conventional vector for each one of these capsids here. So we have a CBA GFP cassette, just a traditional vector genome, and we compared it to AV9, that's a parental serotype. PHPB, which was the capsid that came out of the Gradnaro lab, that's very efficient in mice. And then we had our 2 capsids, AVS and AVS. I'm just going to go back here. This is AVS and this is AVF. We saw that both the F and S capsid produced quite well compared to AAV9, being a little bit better than PHPB at packaging, but very similar. We then went and did just IV injections into C57 Black 6 mice and looked at GFP expression, just native endogenous GFP expression after we did IV injection a couple weeks after. So this is AAV9. This is the whole brain sagittal section. PHPB, as you can see, as reported, gives you really broad transduction of the entire CNS. AVS was very similar to AAV9, but AVS was quite remarkable in its transduction, which, of course, we were very excited about, looked very comparable to PHPB. So a little higher magnification here. You can see across many regions from the cortex, the olfactory bulb, AVF really gave robust transduction. We found that both AAVF and S gave transduction of mainly astrocytes and neurons. This isn't surprising, as AAV after IV delivery really has a high tropism for these cells, and we didn't do any cell-specific selection, so it's not surprising that that's what the capsid that we derived had, that tropism. So we did some quantitation. We saw at a lower dose, and we saw that that AVF had a slightly higher tropism for astrocytes than PHPB, which is more neuronal, and this is reversed in neurons. PHPB had a higher transduction efficiency of neurons and AVF than AVF, but both types transduce both of these cell types and way 100-fold or more better than AAV9. It was also been reported that PHPB does not transduce BALB/C in some related strains, and we really did this experiment to see if the mechanism of transduction was similar. So we compared AAVF in BALB/C mice to PHPB, and you can see that AAVF did not have that same restriction, so it can be used in BALB/C mice as well for gene therapy or gene delivery or tool for scientific studies. So to conclude about this library, it allowed us rapid condensation of the library with this transgene expression after 2 rounds of selection. 1 out of 2 of the tested clones mediated the desired phenotype. Currently, we're testing AAVF in non-human primates to see if some of this phenotype does cross the species barrier. However, we're being realistic. We may need to do direct selection across species either transgenic mice and rats to achieve translation to humans. So I'm going to switch to another gene delivery system that my lab has been developing for over a decade now, and that's the use of extracellular vesicles to enhance gene transfer of virus vectors. So to give you some background of extracellular vesicles, there's many types, and the field is really blossoming in the last decade or so. So microvesicles are usually they bud from the membrane of cells, and they have quite a range, 100 to 1,000 nanometers. There's apoptotic bodies, which are huge, 1 to 5 microns. Those are released from dying cells. There's also these things called exosomes, and those form from these multivesicular bodies, these endocytic structures. And the multivesicular body fuses with the plasma membrane and releases these smaller 30 to 100 nanometer exosomes into the extracellular space. And it's been found that similar to viruses, there's multiple mechanisms of uptake. So it's been found that extracellular vesicles are used in communication between different cells. So these EVs can be taken up by endocytosis, through macropinocytosis, phagocytosis, fusion, many different mechanisms. And a lot of these are similar to how a virus works as well. So in the past several years, viruses and extracellular vesicles are being reported in the same context. Really, if you look back at the virology textbooks that I studied, there was enveloped and unenveloped viruses. There's a clear distinction, and still that's mostly the case. However, there's a lot of literature that's blurring that line. That sometimes a non-enveloped virus at some stage of its life cycle can become enveloped, and often they're using these extracellular vesicles to perform a certain function, such as increased infectivity or immune evasion. So there's examples in the literature, such as with hepatitis virus. It's a classical non-enveloped virus, but this paper showed that it could be packaged into exosomes, and it could evade neutralizing antibodies, and that may be one of the reasons why it can be a chronic infection in some people. Other groups have shown that these enteroviruses, such as rotaviruses and noroviruses, can go into these vesicles, and by going into these vesicles, you can infect with many particles per cell, increasing the infectivity. Even poliovirus has been shown to use a fraction of the virus can use these lipid vesicles to increase infectivity. So in about 2009, we discovered that AAV, again, I told you earlier that's a non-enveloped virus, but a fraction of this virus found in the media of producer cells can be enveloped. And you can see here, this is a cryo-EM, and you can see individual AAV capsids encompassed in this lipid bilayer. So this was a really surprising and exciting finding that we made, and we've been developing the system ever since. So in this study, we showed that you can actually shield the capsid from antibodies and maintain transduction in the presence of these neutralizing antibodies. We also showed that we had an enhanced transduction efficiency with AAV8 and AAV9 when they were associated with vesicles. This could be due to increased trafficking across the blood-brain barrier. We still don't know the mechanism. We've also shown that exosome-associated AAV2 vectors have a really enhanced ability to cross these biological barriers, such as the ILM, after intravitreal injection into the eye. And this is done in mice, but you can see compared to AAV2, we have a greatly increased transduction across the retina. We also showed with a collaboration with Federico Mengozzi that exosome-associated AAV8 had a greatly enhanced transduction of liver, and this was done in the context of a hemophilia B mouse. We also showed in a collaboration with David Corey's group that we could increase transduction of these so-called sensory cells, hair cells, that are in the inner ear. These hair cells are really implicated. They're the target of many therapeutics that are going after hereditary deafness. And we found that these hair cells were were greatly enhanced in the transduction by exosome-associated AAV vectors. There's several other groups that are using exo-AAV as well. In the past, 3 or 4 years, it's been shown that by one group that if you increase this exosome transmembrane protein, you can actually increase the yield of exo-AAV. And there's other groups that have shown increased transduction of the brain and also increased transduction of the retina. It can also be used for cancer gene therapy. So to summarize our work so far on the exo-AAV platform, we've shown increased in vivo transduction compared to conventional AAV, resistance to neutralizing antibodies. So this may be used in patients that have pre-existing antibodies, and so far they have to be excluded from clinical trials. We've shown increased penetration of biological barriers in the brain and the eye. However, this technology is really still in its early days. There's a lot of development that's ongoing to improve the yields, the scalable nature of purification, as well as some regulatory issues that we need to show before it's ready for prime time and moving into human gene therapy. Now with that, I first want to thank you for your attention and for being with me today. And then I need to thank all the great collaborators and scientists that I've had the opportunity to work with over the past several years, all the people that worked in my lab, of course, the funding from the NIH and otherwise that have made this possible. And again, thank you to Biomir for giving me the opportunity to speak with you today. Thank you.
Thank you for an excellent talk, Casey. And let's open up for a couple of questions. So my first question is, the extracellular vesicles definitely have a lot of promise and for non-enveloped viruses to escape immune detection, and there's a lot of fantastic data and work going on. My question is, has anybody shown that using this approach can increase the durable response? So what I mean is, do the exo-AAVs help the virus stay in the target tissue longer and reduce clearance?
No, that's a good question, and I'll try to answer it the best I can. So in terms of AAV is maintained, extrachromosomally, as you know. So generally, there's a low frequency of integration. So the majority of it is going to be in these stable concatenamers, like different AAV genomes joined together in the transduced cell. So as long as there's not a lot of cellular division, those have been shown to be quite stable for many years in preclinical models. Now, with related to exosomes, I don't know if you mean like the immune dampening or immunomodulation. Now that could play a role in terms of if you don't have as much of an immune response to resulting in clearance of transduced cells.
Yeah.
Right now we haven't really looked at that, and we'd have to do a lot more research on that. But certainly there's a possibility that they can be. There's some data showing that the exosomes have some immunodampening effect. They have a lot of phosphatidylserine on their surface, which is actually known to be an immunosuppressant. But again, I think this is going to have to be the result of a lot of work by us and other groups. Yeah.
No, definitely. And I think some folks are using stem cells and things like that to try to, hide viruses and basically get them where they need to go. So I think exosomes make a lot of sense. And it's a really promising approach.
Thank you.
Yeah. So my next question is, when we looked at the gorgeous fluorescence data, what leaps out is, of course, the significant difference in transduction, both, of course, in C57, in the black 6 as well as in the bulb C.
Mm-hmm.
And just for my understand, just for clarity's sake, so the difference between the AAV F and say AAV, the wild type, is really the addition of the 7-mer, right?
You got it. That's it.
And so somehow that's improving blood-brain barrier crossing.
Yeah.
What are your thoughts on how that could be? Because that, again, as you know, is the holy grail. If you can get it into the brain with any degree of efficiency and avoid, stereotactic injections, that would be amazing.
Sure. Yeah. No. It is. It's pretty amazing how these short peptides can cause such a dramatic difference in the tropism and transgene expression. This goes all the way back to phage display. You might have heard of biopanning with bacteriophage. That's really, I mean, AAV folks didn't invent this, right? The libraries have been around with bacteriophage, like I'm saying, and people have used them now in the context of AAV, and it's been shown to give really amazing properties. So now that does have its caveats. You have to understand that you show it in mice, you do it in mice, you're doing a
Yeah.
selection. You may actually, so again, it's a black box approach. So we don't know the mechanism. You can sometimes figure it out. Sometimes it's not that straightforward to figure out. I'll just tell you that we do see, I didn't show the data today, but you do see an increased genomes. We measured genomes across the blood-brain barrier or in the brain. You see an increase. It's not just transgene expression. It's not just there's more GFP, the same amount of virus crosses, a lot more virus crosses with AAVF. So
Okay.
likely there's an increased ability to cross or transcytose the blood-brain barrier. We have no idea how that's occurring because you're just doing a selection approach. You can then try to reverse engineer backtrack, try to figure out how it's working, but we don't know. And that's been shown for PHPB. That's similar. That's how they found that it crossed the blood-brain barrier more because it had that really distinct phenotype in the BALB/C versus the BLAC6. They were
Yeah.
able to identify the receptor that was
Oh,
involved. It was Ly6A.
yeah.
But really, we don't really have a differential phenotype, which really we don't want. We want it to work in many different species. However, again, this is a caveat. We haven't. The testing in non-human primates is ongoing, so we currently don't know if it will be translational. But we think we can use this platform across different species to make it translational in the future.
Right, right. No, definitely. And I think along those lines, one of, again, the key um, caveats and challenge, probably the barriers is that species difference between
Sure.
mice, NHP, and
Yeah,
human.
that's
And
huge.
that is huge, right? Because if it works in mice but doesn't quite translate, that's a bit of an issue. So can you talk a little bit, more about that, how this approach, which is super elegant using the fluorescence tomato mouse approach, do you think there's by making it more efficient, do you predict that you could perhaps improve the translation rate, or is that a bit of a stretch?
I get what you're saying. I mean, first of all, yeah, the species barrier, as you mentioned, is absolutely critical. I mean, we're not making these vectors to work in mice, although they can be great biological tools to study biology, but they're not therapeutically relevant to work for gene therapy in humans. So it's an absolute huge barrier. That said, there are, like, for instance, AV9 works in mice and pigs and monkeys and humans. So, I think just the ability to translate, it's a probability thing, right? So there's a probability that there's a certain probability that you could, I don't know if you could predict it, but it will work in mice and it will work in humans, depending on what pathway
Yeah.
is selected by the virus. But we don't want to have like a low probability, right? We want to have an increased probability. So I think really what most people, including us, are doing is we're going to try to do these selections in non-human primates. So
Yeah.
that's what we're doing now. Now,
Okay.
it's probably not straightforward. You can't make a transgenic monkey that easily, right? With a flock stop, TD tomato
Yep.
reporter.
Oh boy.
But we
Yeah.
do have ways of using another type of strategy that still allows us to do the transgene expression. We're still in development with that. And I think other groups are kind of along the same lines. But I think more people are trying to either go across species. So maybe like mouse, rats, cats, dogs may have a greater chance of being able to translate and maybe going for more conserved mechanism than just going from mouse to monkey. But I think a lot of folks are just going to go right to monkey because it's our closest to
Right.
humans. Yeah.
Yeah. So I'm going to ask an off-the-wall question. So Bob, you may have to edit it out, but this is just me being nosy. So given the cost and given the complexity of working with large animal models, right?
Mm,
Have you guys looked into looking at any kind of organ on a chip or ex vivo? Because, and the reason I asked that is because when I was looking at blood-brain barrier, I know a couple of, well, at least a couple of companies that come to mind have this, basically they have the epithelial cells and then they have the vascular channels, and you can, so it's not as beautiful as a systemic model, but it allows you to very quickly screen what crosses the blood-brain barrier. So basically movement from the vascular to the epithelial, the epithelial layer. And I don't know if it's relevant, but I don't know if that's a more viable option to kind of narrow down your choices before you go into systemic.
You know what? It's a great. I don't think. Yeah, I think this is a great question and a very valid one. And there's many reasons for that. First of all, like as you said, non-human primate work is very expensive. It's also, there's ethical concerns. You don't want to use a lot of animals. So if we can do the work in an ex vivo model, then certainly I think that's always something we should think about. It's just,
Yeah,
are the models good enough now that they
that's
meet
it.
all the biological barriers that
Yeah.
that we have in an animal model? But it is possible that you can combine these organoid models or these 3D culture systems
Hmm.
along with selection to make it relevant. I think we are interested in that. I have a close collaborator, Servio Ramirez at Temple University. He has like a 3D neurovascular unit on these
Wow. Oh,
biofluidic
wow.
chips, and you can
Yeah.
it can flow blood through them. Um,
Yes.
and we are going to be looking at that with these libraries.
Yeah.
Again, there's caveats to every
Of
selection,
course. Yeah.
but I think it's worth it for many of the reasons that I said and that you said. To cross this species barrier.
So thank you, Casey, for a fantastic presentation and definitely sharing your very exciting data. If you'd like to learn more about Casey's research, please visit the lab website, caseymaguirelaboratory.com. Thank you again, Casey.
Thank you so much, Anjali. It was really a pleasure to talk with everybody today. And again, thanks to Biomir for sponsoring this talk, and really look forward to having some people reach out. And thank you very much.
Thank you.